Hi, with this lecture we begin Unit 4 of A&P 241, which is our nervous tissue or nervous system unit. So during this chapter we are going to go over the overview of the nervous system and focus on the histology of nervous tissue as well as the electrophysiology of how action potential gets generated in the neurons and travels or gets conducted through the length of the neuron, as well as jumps from one neuron to the next neuron. We are also going to talk about different neurotransmitters. So neurotransmitters are substances that carry action potential in that space between one neuron to the other neuron, which is called a synapse. To end the chapter, we're also going to talk about the different neural disorders. So in part 1, we are going to talk about the overview as well as the histology. In part 2, I'm going to talk about the physiology and the synapses and the neurotransmitters. So in terms of body weight, the nervous system does not take up a lot. It only takes up about 3% of the total body weight. The subcategory of biology that deals with the nervous system is called neurobiology, and you would see a neurologist when you have disorders of the nervous system. If you have to have surgery of the nervous system or the brain, you would see a neurosurgeon. Now, the structural and functional unit of the nervous system is a neuron, which is also known as the nerve cell. Now, we are born with all the neurons that we are going to have for the rest of our life. So neurons are cells in the human body that do not multiply by mitosis. We have already talked about some terminology when it comes to the nervous system throughout A&P 241. Now, any signal, when I talk about the signal, I'm talking about the signal from the sensory receptor inwards towards the central nervous system. Now, any signal that goes towards the central nervous system is referred to as an afferent, with an A, is referred to as an afferent nerve signal. Any signal that comes outwards from the central nervous system is called an efferent signal, efferent with an E. Now, wherever the efferent signal ends up, so whatever organ carries out the response is referred to as an effector organ. So the effector organ is whatever organ that the efferent signal ends up at. The overall function of the nervous system is to regulate the body. It's the command center of the body. So in turn, what it's essentially doing is maintaining the overall homeostasis of the human body, internal coordination. And it does that using chemical messengers via the endocrine system. If you recall, the endocrine system, anything that's endocrine, is when the substances or hormones are secreted directly into your bloodstream. Now, the pituitary gland, which is located in the brain, and we'll look at the pituitary gland when we talk about the structure of the brain. Now, it regulates the endocrine system, and the endocrine system does that using, one, the sense organs to sense and determine what is to be done. So that is referred to as its sensory function. So it senses whether homeostasis is present, whether it's disrupted, so it has to first sense what is going on. Now, once it senses, it has to determine what its response is going to be, or whether a response is needed in the first place. So the two organs that constitute the central nervous system are the brain and the spinal cord. So this constitutes the integrative function. So the integrative function determines whether a response is necessary and what that response is going to be. And then it issues commands to the glands or the muscles, which are going to be your effector organs. And that part is called its motor functions. So the nervous system has three functions: the sensory function, integrative function, and motor functions. The sensory function is to 1. Receive information through its sensory receptors. Integrative function is when it decides if a response is necessary and what the response is going to be. And 3, it's motor function, where it's sending a response to its effector organs. Now, the nervous system has a few subdivisions. The two major subdivisions are the central nervous system and the peripheral nervous system. In simple terms, the central nervous system is the brain and the spinal cord. The functions of the brain and the spinal cord are integrative functions. So these two organs basically decide whether a response is necessary and what the response is going to be. So the function of these two organs is integration. So the brain is located in the cranial cavity. The spinal cord is located in the vertebral canal. We have two separate chapters for these two organs, for its structure and its physiology. The peripheral nervous system is basically anything that is not the brain and the spinal cord. So the peripheral nervous system consists of nerves that originate from the brain, nerves that originate from the spinal cord. You have certain structures called ganglia, plural, or ganglion in singular. So what these structures are, they are little lumps of neurons or cell bodies. Then you have little braided structures of nerves called plexuses. And the peripheral nervous system consists of sensory receptors. For example, we've already talked about certain sensory receptors that are located in your integumentary system. So Purkinje corpuscles, Merkel cells, those are all examples of sensory receptors. So here's a nice flowchart that talks about the further classification of the peripheral nervous system. Now, the peripheral nervous system reports back to the central nervous system. So this beige box is the CNS, and this purple box is the PNS, or the peripheral nervous system. Now, the peripheral nervous system is further subdivided into three subcategories. Let's take them one at a time. So on the far left, you have the somatic nervous system. Now, the somatic nervous system consists of voluntary functions. So under the somatic nervous system, you have the somatic sensory division as well as somatic motor division. So under the somatic sensory division, you have the sensory receptors that are located in the integumentary system. Under the somatic motor division, you have motor neurons that are located in the skeletal muscle. So skeletal muscles are your voluntary muscles. So the somatic nervous system deals with neurons that are located in your voluntary organs. The next subdivision is the autonomic nervous system. Now this subdivision deals with receptors and neurons located in your involuntary organs. So the autonomic sensory receptors are located in organs of your body that are completely involuntary. So you would have these sensory receptors in your blood vessels, in your heart, in your intestines, so in parts of your body that are beyond your control. Autonomic motor neurons are located in your smooth muscles and are further divided into sympathetic and parasympathetic divisions. We have a complete chapter on the autonomic nervous system. So this division, the autonomic nervous system, deals with involuntary parts of your body. The third division of the peripheral nervous system is the enteric nervous system. anything that is enteric has to do with your GI tract. So the enteric nervous system also deals with involuntary parts of your body, which the GI tract is, but it specifically deals with your the smooth muscles and the receptors in your gastrointestinal system. So to recap, there are 3 subdivisions in the peripheral nervous system. The somatic nervous system, which deals with the sensory receptors and the motor neurons located in the voluntary parts of the body. The autonomic nervous system, which deals with the sensory receptors and the motor neurons in the involuntary parts of the body. And then you have the enteric nervous system, which specifically deals with receptors and motor neurons in the gastrointestinal tract. So we already talked about this. Just goes over what I just talked about. Somatic receptors in the skin, specifically the skeletal muscles, the bones, and the joints, which are all voluntary. Autonomic goes over all the visceral organs, which are all involuntary. And enteric goes over the GI tract. Now, when we talk about nervous tissue, we have to discuss its structural and functional unit, which is a neuron. Now, the neuron is a special cell, one, because it cannot reproduce by mitosis, and it has 3 exclusive properties. One is the property to be excitable. It responds to stimulus. It can generate action potential. It can produce electrical signals. 2, it can conduct electrical signals. It can transmit electrical signals through the length of itself. 3, it can secrete chemical substances called neurotransmitters. We will talk about the different types of neurotransmitters at the end of part 2 of this chapter. Now, in nervous tissue, we don't just have neurons. Neurons are the major structural and functional units of nervous tissue, but in addition to neurons, we have support cells to the neurons. Now, those cells are called neuroglia, and we have different types of neuroglia. So let's look at a structure of the neuron. You do need to know the entire structure of the neuron in its entirety. So a neuron basically looks like a comet. It's got a star-shaped body, and it's got a long tail at the end. It's got a long tail at the end that ends in shorter tails. So that's the tail right there. So the body of the neuron is referred to as the soma, or you can just call it the cell body. So in the cell body, you have all of your basic cell organelles. So you have your nucleus, you have the mitochondria, you have the smooth endoplasmic reticulum, the rough endoplasmic reticulum. You have all of those cell organelles. Now, in a generic neuron, arising from the cell body are these little structures that look like branches. So I'm shading them right now in green. So all these little branch-like structures, and there are a variable number of these, are called dendrites. So the cell body, or soma, has the nucleus. It has all of these structures, branch-like structures called dendrites, arising out of them. Now, in the cell body, you have the lysosomes as well. Now, anytime the lysosome of a neuron breaks down something, it's very hard to get rid of it. Now, whenever a lysosome of a neuron breaks down a foreign body or a worn-out cell organelle, there's a byproduct that remains within the neuron and it's hard to break down. This byproduct is called lipofuscin. Now, as we age, the amount of lipofuscin increases, and it may contribute to a condition called macular degeneration. Macular degeneration is when the retina breaks down and causes blindness. So when we talk more about these branch-like structures called dendrites, they are structures that receive information from neighboring neurons. So a neuron does not exist solitarily. So you have other neurons in its vicinity that are transporting or transmitting action potentials. So the dendrites are receiving or accepting information or action potentials from neighboring neurons, and the dendrites are accepting these informations. Now, these granulated substances that you see on dendrites are receptor proteins. They're receptor proteins that kind of accept external chemical messages. Now, the difference between a dendrite versus an axon. So now, what's an axon? The axon is the longest protrusion or projection of a neuron. So the long tail that you see, this guy right here, the long tail is the axon. The difference between a dendrite and an axon is the length. Dendrites are numerous and much shorter than an axon. Now, typically, in a generic neuron, there is only one axon. But you have neurons with more than one axon as well, and we're going to talk about those variations in just a little bit. Now, in a generic neuron, you have one axon, and it begins at this conical region called the axon hillock. The axon hillock is present on the cell body or the soma. Now, the axon contains cytoplasm, but it's called the axoplasm, and the plasma membrane covering the axon is called the axolemma. Now, the axon may have branches, it may or may not have branches, and they are called axon collaterals. Now, in this particular image, you have one axon collateral right here. At the very end of the axon, you might have additional branches. There are several additional branches in this axon. Here's a branch, here's a branch. And at the end of each branch, you have a little bump. Here's a little bump, here's a little bump. Now, each of these bumps is called a synaptic end bulb. Now, this synaptic end bulb is going to be extremely important when we discuss synapses. A synapse is a junction between one axon and another axon, or one axon and an effector organ. Now, the synaptic end bulb is where a neurotransmitter is housed. So this is where the action potential is going to jump from one axon to another neuron, or one axon to another effector organ. So to put things in perspective regarding the size of a neuron, the cell body of a neuron could range anywhere between 5 to 135 micrometers in diameter. So just to clarify, 1 micrometer or 1 millimeter is 1,000 micrometers. And an axon could range anywhere between 1 to 20 micrometers in diameter, which is its cross section. Now, how does stuff travel from the cell body to the synaptic end bulb, and from the synaptic end bulb back to the cell body. Now remember, all the cell organelles, the nucleus, the lysosomes, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, all of that stuff is only located in the cell body. It is not located in the axon. So anything that's secreted, worn out organelles, proteins, anything that's supposed to be made has to be made in the soma or the cell body. And it has to be transported across this very large cell. And it happens in two ways. Anytime the axolemma has to be repaired, channel proteins, enzymes, anything that has to be disposed of, it has to be transported back and forth. And it's transported with the help of the cytoskeleton. Now, if you recall cytoskeleton back from our first 3 chapters, it is made up of 3 components: the microtubules, the microfilaments, and the intermediate filaments. So it happens with the help of the cytoskeleton. So let's look at the 2 ways that axonal transport takes place. There are 2 mechanisms for axonal transport: the slow axonal transport versus the fast axonal transport. So slow axonal transport is only reserved for large compounds such as the axoplasm. So it's basically for routine maintenance items. So axoplasm, which is the cytoplasm inside of the axon. So it's for routine maintenance. It only happens in one way. So it happens away from the soma or the cytoplasm, down the axon. So it happens anterograde, down from the soma. And it travels about 1 to 5 millimeters per day. So it's very, very slow. Hence, it's only reserved for routine maintenance. Fast axonal transport cannot afford to happen that slowly. It happens about 200 to 400 millimeters per day. And it's meant for damaged organelles, proteins, enzymes, pathogens. And this method of transport can happen both ways. It can happen from the cell body towards the synaptic end bulb, and it can happen from the synaptic end bulb back towards the cell body. So it's called fast anterograde transport, which is down the axon. It's meant for organelles, enzymes, vesicles, and small molecules. Or it can happen fast retrograde, which is from the synaptic end bulb towards the cell body. So just picture this happening. So it goes both ways. It goes up the axon. So it goes downstream as well as upstream. So if any pathogen, so if someone was to get meningitis or an infection of some kind, it happens both ways. So fast axonal transport is reserved for those instances wherein the body has to handle these things fast and quick. Now let's talk about the variations in neuron structure. Earlier we looked at the basic structure of the neuron. Now we're going to talk about the variations in neuron structure. We saw the cell body, we saw the dendrites, we saw the axon hillock, and the synaptic end bulbs. Now we're going to talk about the 3 variations in structure. The most common neuron that we talked about earlier is the multipolar neuron, where it has several dendrites. Now what I want you to look at when you identify these is how many projections are exiting out of the cell body. So right here to the left, you have a multipolar neuron. This is the cell body, and you have numerous projections sticking out of the cell body. This is what makes the neuron multipolar. These are the most common types of neurons in the body. The next type of neuron is a bipolar neuron. And these are common in the sensory organs. So olfactory, which is smell, retina, which is the eye, the ear. And if you look at this neuron, there are only two projections sticking out of the cell body. Finally, you have the unipolar neuron, which are found in some sensory receptors of the general senses, such as the touch, nociception, which is pain. So here's the cell body. You only have one projection sticking out of the neuron. Now, some structures that we did not go over are these sausage-like coverings on the axon. These sausage-like coverings on the axons are called the myelin sheath. Now, neurons may either be myelinated or unmyelinated. If there's no myelin sheath, the neuron is called an unmyelinated neuron. If there's myelin sheath, it is called a myelinated neuron. Now, these gaps between the myelin sheath are called nodes of Ranvier. My apologies, I forgot to mention this in the previous slide where we actually discussed the structure of the neuron. The gaps in the myelin sheath are called nodes of Ranvier. When we talk about conduction of action potential, we will go over the importance of the nodes of Ranvier. Now, based on function, there are three functions of neurons. A neuron can either be a sensory or an afferent neuron, meaning that a neuron can synapse with a sensory receptor and can carry an afferent signal. So it can detect changes in the body or in the environment and can transmit this information to the central nervous system. These neurons are mostly unipolar, meaning they only have one projection out of the cell body. Now, any neuron that is present only in the central nervous system, so a neuron that is part of only the spinal cord or the brain, is called an interneuron. Now, this neuron, an interneuron, is almost always multipolar. So it has several dendrites and axon terminals. Now, these neurons serve as information highways. They're in the brain, they process energy, they integrate, they store, they retrieve informations, and so they're supposed to make decisions as to what kind of response is meant to be sent out. So they're decision-making neurons. And then lastly, you have the motor neurons or the efferent neurons, which send out the efferent signal to the effector organ. So think about this. A motor neuron always goes to a muscle or a gland. So it always goes to a muscle or a gland of some kind. Whenever you see the term motor, it always goes to a muscle or a gland. Now, this muscle can either be a voluntary muscle or an involuntary muscle or a cardiac muscle. But motor means it is going to a muscle. These are, again, multipolar neurons. Now, let's see what happens. So let's understand this diagram. So neurons or nerves are part of the peripheral nervous nerves, not neurons, because neurons can also be part of the central nervous system. I should say nerves are part of the peripheral nervous system. Brain and spinal cord are parts of the central nervous system. So here you have a sensory neuron. So let's see, right here is a sensory neuron. So this sensory neuron is present in a sensory receptor in this person's skin. So for example, if this person has a pinprick, what is the sensation that you feel when you have a pinprick. It's pain. So when this person got pricked with a needle, they felt pain. So this sensation, which is pain, got sensed by this person's sensory neuron. The signal via an action potential got transmitted by this unipolar neuron. Why is this neuron unipolar? Because here's the cell body. It's got one projection coming out of it. The sensation got carried, this afferent signal got carried by this unipolar neuron, and it got sent to this, now we're in this blue shaded box, so we're in the central nervous system. So here's a synapse. This little neuron is only present in the central nervous system. Hence, this neuron is called an interneuron. It's called an interneuron because it's only present in the central nervous system. Now, this neuron is going to make a decision. This neuron is going to make a decision and send out an efferent signal. Now, an efferent signal is carried out through a motor neuron. So here's a motor neuron right here, and it's being sent out to this person's muscle right here, which is a skeletal muscle, and this person is going to pull their arm away from the needle. Now, the next concept that we're going to talk about is the neuroglia, also referred to as glial cells. Now, in the human body, we have about 1 trillion neurons, which are extremely important to the human body. However, we have about 50 trillion neuroglia or glial cells. Now, glial cells are just as important to the nervous system as neurons are. So let us talk about what these glial cells are and how they help the nervous system. We have different types of neuroglia in the central nervous system versus the peripheral nervous system, and you do need to know the names and the functions of each type of neuroglia. And so first, let's look at the glial cells of the central nervous system. There are 4 types of glial cells in the central nervous system. The first type are called oligodendrocytes. So remember, we're in the CNS right now. The first type of glial cells are called oligodendrocytes. Oligo refers to lipids. Oligodendrocytes are present on the axons and form myelin sheaths. Myelin sheaths are extremely important. They form an insulating sheath around the axon and prevent the action potential from leaking. The next type of neuroglia are called ependymal cells. Now, ependymal cells are cells that produce cerebrospinal fluid. The next type of cells are called microglia, and microglia have the macrophage lineage, so you already know what they're going to do. They're going to phagocytosize dead cells, foreign matter, and microorganisms. So these are also extremely important because the lack of microglial cells will result in a lot of toxins not being filtered out through your cerebrospinal fluid. And the last and the star players, literally the star players of your neuroglia are astrocytes. Now, astrocytes are the most abundant type of neuroglia because, one, they form the structural framework of the CNS by forming the blood-brain barrier. They form the blood-brain barrier by forming tight junctions between the neurons. If you recall back to the tissues chapter and the cell junctions, they form tight junctions by forming the cadherin zipper. They also convert glucose to lactate. Now they ferment. If you remember the glycolysis and the Krebs cycle, In the brain, glucose actually undergoes glycolysis, but instead of going through the Krebs cycle, it undergoes fermentation and gets converted to lactic acid, which is used to provide nutrition to the neurons. They also secrete nerve growth factor. And when lactic acid is present, action potential transmits fastest and most effectively. Now, any and every types of brain tumors, because neuroglia do divide by mitosis, brain tumors of any and every neuroglia is possible and happens. However, they are more common in children than they are in adults. The glial cells of the peripheral nervous system are only two. They are Schwann cells and satellite cells. Schwann cells myelinate the axons in the peripheral nervous system, and satellite cells provide and regulate neural cell bodies and fluid. So there's no tight junctions and that kind of stuff in the peripheral nervous system. So they're basically just maintaining nerves. So they don't have to form tight junctions, the blood-brain barrier, and all that kind of stuff. So that's only in the central nervous system. Now, talking a little bit about the myelin. So the myelin, so let's go over the structure of the neuron one more time. So you know this is the cell body, these are the dendrites right here, this is the axon hillock, this is the axon collateral, the axon itself. These sausage-looking things are the myelin. The gaps right here are the nodes of Ranvier. These are the axon terminals and the synaptic end bulbs. Now, the rough endoplasmic reticulum of the neuron is called a Nissl body, or Nissl granules. You'll find the textbook refer to it as Nissl granules as well. They're large because they're secreting a large amount of protein, because your neurotransmitters are nothing but proteins. Now, the myelin is basically a lipid and a protein-based substance that, one, insulates the axon, and two, it increases the speed of action potential that is going to travel in this direction. So down the length of the neuron. So action potential is going to travel down the length of the neuron, and the myelin is going to increase the speed of that. In the CNS, the central nervous system, the oligodendrocyte is synthesizing it. In the peripheral nervous system, it is the Schwann cell that is synthesizing it. The myelin sheath, the composition of the myelin sheath is pretty similar to that of the plasma membrane phospholipid bilayer. Now here's a lovely transmission electron micrograph cross-section or transverse section. So this blue substance here is an axon. This brown layer here is the myelin. This whole thing is a Schwann cell, so that tells you that this is from the peripheral nervous system and not the central nervous system. This is the nucleus of the Schwann cell, and this is the cytoplasm of the Schwann cell. Now, the way the Schwann cell lays down this myelin is that it creates spirals. So you can see here in this illustration how it's laying down the myelin sheath in a spiral fashion. So this is how it lays down the myelin sheath. Now the oligodendrocytes, because this is happening in the central nervous system, they myelinate several fibers, and by fibers we're talking about axons. They myelinate several axons at once instead of one at a time. And there's no neural lemma. So here's a nerve fiber, here's an oligodendrocyte. So this one oligodendrocyte here, is doing the job of myelinating 1, 2, and 3 separate nerve fibers at once, instead of just doing one at a time. Now, in the peripheral nervous system, you also have parts of axons that are unmyelinated. So these could be dendrites, these could be axon terminals. Now, Schwann cells can skip areas by having little grooves. So what you're seeing here in this transmission electron micrograph on the right is, here's the nucleus of the Schwann cell, and here's little areas. This is an unmyelinated chunk, here's an unmyelinated chunk. So the Schwann cell has basically just skipped areas where it hasn't myelinated. So it's just skipped wrapping that part with myelin. Now, in the peripheral nervous system, you have white matter versus gray matter. Myelin, because it's a lipid-based substance, appears to your eye a white-colored substance. Anything that is unmyelinated or cell bodies appear gray in color. So that is why when we look at nervous tissue, we call it gray matter and white matter. So when you look at cadaver tissue, you have to know the differences between gray matter and white matter in the brain versus the spinal cord. So in the peripheral nervous system, a nerve is a bundle of neural axons. Ganglion is a cluster of cell bodies. Now in the central nervous system, you have gray matter and white matter. So to the left, you're seeing a transverse section or a cross section of the spinal cord. So spinal cord, horizontal section to the left. The gray matter is present in an X fashion towards the center, and the white matter is present towards the periphery. We'll talk more about this when we get to the spinal cord chapter. To the right, you are seeing a frontal section of the brain. You should remember all your planes from our very first anatomy lab where we talked about the different planes of the human body. So you're seeing this plane right here. So in the brain, you see white matter towards the central portions and gray matter towards the periphery. Now this is setting down a foundation for what I'm going to cover in the next part of this chapter, neural communication. Now whenever an action potential or a signal is going to happen, there are two types of electrical signals. One is referred to as a graded potential. So what a graded potential is short distance communication. Now, at any given point, you are exposed to a lot of stimuli. Not every stimulus is going to generate a response. So a short distance stimulus is a stimulus that goes from the dendrites over the cell body to the axon hillock. So just try to imagine this. So it goes along the dendrites. I'm looking for my pointer here. It goes along the dendrites and the somas. So short distance stimulus. If the stimulus is large enough, the graded potential can generate an action potential. So an action potential is long distance communication. So if a stimulus is large enough, so say right now as you're sitting in this room, you feel air hitting on your face. That stimulus is probably not large enough for you to actually respond. It would be a short distance communication. It's a graded communication. But if I was to make a loud sound that is very uncomfortable for you, and it's likely to get you to respond, that graded potential would convert into an action potential. So that action potential would not just go along the dendrite and soma, but it would go down the axon and stimulate the release of neurotransmitters from the synaptic end bulb and get a response. So that is called an action potential. So in contrast, a graded potential is a short distance communication and action potential is a long distance communication. We're going to look at exactly what that happens in the neurophysiology, the actual chemistry of what that happens in the next chapter. So here is an overview. So look at this diagram right here. What happens in this pathway where you are making a thought, you are thinking, you're consciously making a thought about writing? So when you touch the pen, you are touching the pen, a graded potential happens. So in point number 1, I'm looking for my pointer here, right here. In point number 1, you touch the pen. So you have a sensory receptor in the skin in your finger that initiates a sensory receptor. So the sensory receptor is initiated, a graded potential is started in the axon. A graded potential travels along the dendrites, travels along the axon, initiates an action potential, an afferent signal is initiated. The blue line is your afferent signal. It goes along your sensory neuron. It goes here. You're still in the peripheral nervous system. Follow the line. You're now in the spinal cord. The purple line is an interneuron. Now the interneuron has decided that you want to write. The neurotransmitters in the interneuron have decided that an action potential is going to be initiated, and the red neuron here is going to be a motor neuron. So a motor neuron is sending an efferent signal down to your effector organ, which is going to be the muscles in your finger, and you are going to be able to move the muscles in your finger, and that is going to be enabling you to write. Now, electrical potential. When we talk about electrical potential or action potential, we are talking about charged particles. So electrical potential happens because your axolemma, think about the term axolemma. This is going to be a lot of chemistry here. So axolemma is basically the plasma membrane of your axon. So you have the cytoplasm inside of an axon, you have the extracellular fluid of an axon. So there is potential energy inside this axolemma. You have more negative charge inside and more positive charge on the outside. And I will explain why this is in a separate video. Now this results in potential energy being present in the axolemma. This is called resting membrane potential. So point to note, the point to think about before you watch my next video is that the resting membrane potential is negative 70 millivolts. Unless you mention the term millivolts, just the number negative 70 does not mean much. So living cells are polarized. We're polarized because the charge inside of our cells is more negative than the outside. And this is because our DNA, our proteins, are more negatively charged. This is why our resting membrane potential is negative 70 millivolts. So in the next video, I will explain in detail why this is.